Howdy, I'm Alejandro Jenner, but you can call me Alex. I'm here to bring you to the finish line in the last section of biochemistry. Metabolism encompasses the various processes that contribute to a cell's energy requirements and waste removal. It involves the creation of and interconversion between 4 different classes of macromolecules: the carbohydrates, lipids, proteins, and nucleic acids that make up our cells. Everyone learns a little differently, but please, grab a notebook and draw the pathways as I cover them. As we work together, I'll emphasize frequently tested enzymes and concepts to help you max out on test day. So, let's get going. It's useful to group metabolic reactions by the site in which they occur. Most reactions occur in either the mitochondria of a cell or the cell cytoplasm, although some reactions occur in both. Don't be surprised if a USMLE question asks where a certain reaction takes place. So please, know these. In the mitochondria, reactions such as fatty acid oxidation, otherwise known as beta oxidation, acetyl-CoA production, the TCA cycle, otherwise known as the Krebs cycle or the citric acid cycle, oxidative phosphorylation, and ketogenesis take place. Side note, the guy that discovered the citric acid cycle, Adolf Krebs, won the Nobel Prize in Medicine and Physiology in 1953. It won't show up on your exam, but it's a good idea to know who won Nobels in medically related fields, because some of the folks that write USMLE questions trained with and are friends with some of these laureates. The cytoplasm plays host to glycolysis, fatty acid synthesis, and the hexose monophosphate shunt, protein synthesis in the rough ER, steroid synthesis in the smooth ER, and cholesterol synthesis. Do you remember which types of proteins are produced in the rough ER? Proteins that are meant to be secreted from the cell, bound within organelles, or embedded in membranes are all synthesized on the rough ER. Steroid and cholesterol synthesis occur in the smooth ER. Which reactions take place in both the mitochondria and the cytoplasm? These include heme synthesis, the urea cycle, and gluconeogenesis. An easy way to remember these is that it takes 2 to hug. That said, if you look closely, you'll see that parts of these pathways occur in one location, while the rest occur in a separate compartment. By the way, do you remember what vitamin is required for heme synthesis? That'd be vitamin B6, or pyridoxine, which is used to convert succinyl-CoA and glycine into aminolevulinic acid, the first step in making heme. The first step is usually, but not always, rate limiting. Speaking of steps, you'd think that these metabolites are studying to be MDs or something. Okay, flash quiz time. Let's see if you're paying attention. Glycolysis occurs in the blank, either mitochondria or cytoplasm, whereas the TCA cycle occurs in the blank, either mitochondria or cytoplasm. I'll give you a second. It's important to know where these pathways occur. Glycolysis occurs in the cytoplasm, whereas the TCA cycle takes place in the mitochondria. Before we talk about a bunch of specific enzymes, we need to learn some terminology first. The name and function of the enzyme are often related. If you understand this nomenclature, you can usually figure out the basic function of an enzyme. Let's briefly cover some of the more important enzyme categories here. We start off with our first category of enzymes, the kinases. These enzymes transfer a high-energy phosphate group from ATP to a target substrate. An example of a kinase is phosphofructokinase. You'll find phosphofructokinase in the glycolysis pathway, where glucose is broken down to create ATP. Phosphofructokinase phosphorylates or adds another phosphate to fructose 6-phosphate in glycolysis. Notice how the phosphate group is taken from ATP and moves to the first carbon of fructose 6-phosphate. What do you think a phosphorylase does? Phosphorylase enzymes are similar to kinases, meaning they add high-energy phosphate groups to a substrate. But instead, this set of enzymes do it without using ATP. An example of a phosphorylase would be glycogen phosphorylase, which is found in the glycogenolysis pathway. Glycogenolysis is where you take a large glycogen chain and break off free glucose molecules. Glycogen phosphorylase grabs a free phosphate and cleaves the 1,4-glycosidic bonds in glycogen to produce a glucose 1-phosphate by adding an inorganic phosphate to it. Phosphatase enzymes are the opposite of kinases and phosphorylases. Phosphatases, like fructose 1,6-bisphosphatase, remove phosphate groups from a substrate. Fructose 1,6-bisphosphatase converts fructose 1,6-bisphosphate to fructose 6-phosphate in gluconeogenesis. Here, we saw a substrate with 2 phosphates attached, and after enzymatic removal, the product has only 1 phosphate, so the phosphatases cleave off a phosphate group. Dehydrogenases catalyze oxidation-reduction reactions. Do you remember what a redox reaction is? It's the same thing. It means that hydrogens and their corresponding electrons are removed from one substrate and added to another. An important example that we'll cover is pyruvate dehydrogenase, which converts pyruvate into acetyl-CoA. Here, we see pyruvate dehydrogenase moving electrons and protons to form NADH, which we'll use later. Pyruvate carboxylase, a carboxylase, moves carbon dioxide groups onto substrates, as you can see here. A pyruvate molecule has a CO2 group added on with the help of pyruvate carboxylase to make oxaloacetate, a molecule with two CO2 groups, one attached by the enzyme. The carboxylase enzymes need the help of biotin, otherwise known as vitamin B7. Whenever you hear the word carboxylase, think about biotin. What would you call the set of enzymes that remove CO2 groups? Hopefully you weren't thinking too hard. It's the decarboxylases that remove CO2 groups. Enzymes that add hydroxyl groups to substrates are called hydroxylases. And lastly, mutases are enzymes that move functional groups around from one side of a molecule to another. An example of a mutase would be methylmalonyl-CoA mutase. How many substrates does a mutase act on at once? Well, it's kind of a trick question, right? Because it's just one. Remember, it moves groups around, compared to the others that we've mentioned that produce a net gain or loss of a functional group. Understanding the naming of enzymes will help you understand the different metabolic pathways in the body. We have many different processes at our disposal, and when we have a dysfunction in one of these processes, we can get different diseases. Okay, one thing we gotta know is the rate-limiting enzymes. These are the most commonly tested enzymes, since they determine the rate at which reactions can occur. But please, don't just memorize these ones though, because it's quite common to find questions about enzymes immediately upstream or downstream of these most commonly tested ones. The rate-determining enzymes are also the ones most commonly targeted by drugs, because slowing them down or speeding them up will have an effect on the processes they're involved in, which might not be the case for other enzymes in the pathway. It'll definitely help you on your test to know which enzyme is rate-limiting for each process, and how the enzyme is regulated. For example, HMG-CoA reductase is the rate-limiting step in cholesterol synthesis. HMG-CoA reductase converts HMG-CoA to mevalonate, which eventually gets turned into cholesterol. Do you remember which class of drugs inhibits this enzyme? It's the statins, which are used to treat hypercholesterolemia. The statins inhibit the rate-limiting step for cholesterol synthesis. We'll cover all of these in a little bit, so I won't read them off to you now. However, you might want to come back to review them later, maybe by using flashcards. We'll go over how each enzyme is regulated in detail throughout the chapter. So take a breath. If you're following along in the book, the summary of pathways diagram may seem a little bit intimidating, but it's meant to show you how all of the metabolic pathways that we're about to go over are interconnected. We simplify it here on the slide to make it a little easier to interpret. The take-home message from this is that carbohydrate metabolism is connected to protein metabolism, lipid metabolism, and even nucleic acid synthesis. In this figure, just recognize how everything is interrelated. We'll talk about each of these processes throughout our lecture today. Once you feel comfortable with the various smaller pathways that we'll discuss in the upcoming slides, it'll be helpful to look back here and see how it all fits together. We'll slowly introduce enzymes, substrates, and products that'll fill this box diagram into a more complete picture. This is also a great page in your book to take notes from other parts of the chapter, or even from other chapters, since there are lots more inputs and outputs to this diagram that are shown here. For example, 1,3-bisphosphoglycerate is used to make 2,3-bisphosphoglycerate, which is used in erythrocytes to reduce the affinity of hemoglobin to oxygen, and will be discussed more in the respiratory chapter. Also, fatty acids are broken down into acetyl-CoA, which is transported using the carnitine shuttle to the mitochondria, where they're converted to acetyl-CoA and can then enter the TCA cycle. As you study, you'll probably find lots more things to add to this page. For now, don't feel like you need to memorize all of this before moving on. Just realize that this diagram in the book should make a little more sense after we break the pathways down. You should eventually be comfortable enough to reproduce the main parts of this image from memory fairly easily. So a little cliche, but hey, practice makes better. We'll start to deconstruct the elaborate summary of pathways diagram by first talking about energy production. What's the main energy molecule the body needs? Hopefully you remember about adenosine triphosphate, or ATP. which stores energy via a high-energy phosphate bond. The body has multiple processes to create ATP depending on whether oxygen is present or not. Generally, when oxygen is present, it's called aerobic metabolism, and when oxygen is absent, it's called anaerobic metabolism. Aerobic metabolism takes place in both the cytoplasm and mitochondria, where oxygen is the final electron acceptor. Aerobic metabolism of glucose can produce 32 ATP molecules via the malate aspartate shuttle found in the heart and liver. This shuttle transports NADH electrons gathered in the cytoplasm to the mitochondrial NADH acceptor, which can produce about 3 molecules of ATP per NADH. However, only 30 molecules of ATP are produced when the glycerol 3-phosphate shuttle is used in muscle. The difference in this shuttle is that it transports NADH electrons from the cytoplasm to the mitochondrial FADH2 acceptor, which can only produce about 2 molecules of ATP per NADH consumed. Anaerobic ATP production takes place only in the cytoplasm of cells, and does not require oxygen. In what cell type of the human body is anaerobic metabolism the only source of ATP? I'll give you a hint. Which cell type, when mature, doesn't have any mitochondria in it? It's the red blood cells. There are some other cell types in the body that heavily rely on anaerobic metabolism too, but more on those later. Anaerobic ATP production, or glycolysis, produces 4 ATP, but requires 2 ATP to be invested. So actually, anaerobic metabolism has a net gain of only 2 ATP molecules per glucose. ATP can also be manipulated and hydrolyzed in otherwise energetically unfavorable reactions that would not occur spontaneously, to provide the necessary reactant for the reaction to occur. Remember I said that each process in the body has a certain function. For example, the function of glycolysis, the TCA cycle, and the electron transport chain are to produce large amounts of ATP for the body. Fun fact: arsenic poisoning disrupts glycolysis by inhibiting the rate-limiting enzyme pyruvate dehydrogenase. So arsenic will inhibit the body's ability to create most of its ATP. When a cell is starved for ATP, or energy, it may eventually die. If it helps you, try to remember the silly phrase, "Don't be an arsenic," along with the inhibition of pyruvate dehydrogenase in glycolysis. Okay, flash quiz time. A 25-year-old runner is exercising to the point of requiring anaerobic glycolysis. How many ATP per glucose molecule does she produce? I'll give you a bit. In the case of pure anaerobic metabolism, she'll make 4, but only a net of 2 molecules of ATP are produced for every molecule of glucose consumed. This is dramatically less than 32 ATP produced by the malate aspartate shuttle when oxygen is available for aerobic metabolism.